Welcome back to another video tutorial. This is Rajesh PALAKURTHI. Now we are going to be discuss about the topic is the DNA, the chapter of molecular basis of inheritance. Okay, the DNA. Yes, we know the full form of DNA. We know the general features of the DNA. Already we discussed in previous class. Now we have to discuss about some little bit more about the DNA and what is the length of the DNA. How the DNA is made up of different kinds of subunits, what they are called, how the every strand of the DNA is synthesized. Everything we will discuss in this chapter. Now see the DNA. Here DNA is biomacromolecule. The DNA is biomacromolecule. It is one type of biomolecule, but we know that biomolecules are two types: biomicromolecules, biomacromolecules. Already we discussed about the two type of the fractions in biomolecules chapter: acid soluble fraction, acid insoluble fraction, or retentate fraction. The molecules which are filtered and which are present in acid soluble fraction, they are called as biomicromolecules, but which are remains in acid insoluble fraction, they are called as biomicromolecules. Here DNA is one type of example of biomicromolecule. Along with the DNA, proteins also macromolecules, and some lipids also macromolecules, polysaccharides also macromolecules. Okay, now our topic is the DNA. Yes, DNA is biomacromolecule. Now it is that's why it is found in which pole? Acid insoluble fraction. Okay, DNA is a long polymer of deoxyribonucleotides. Polymer means the structure which can made up of many monomers. They are called as a polymer. Here the DNA is what? Long polymer of deoxyribonucleotides, means every strand of the DNA, among the two strands, each strand of the DNA is made up of many nucleotides. What are the nucleotides? About the nucleotides we will discuss in the next topic. Yes, now what we are reading here, what we are saying here, every strand of DNA is made up of many nucleotides. Okay, but DNA is acidic in nature and present inside the nucleus. Generally the DNA, the genomic DNA, present inside the nucleus in all living organisms. Okay, right. The DNA was first identified by the scientist Frederick Mischer. What is the name of the scientist? Frederick Mischer, in 1869, in the nucleus of pus cells, WBC. For the first time, DNA is identified in which cells? Nucleus of white blood cells. By which scientist? Frederick Mischer. But Frederick Mischer is not says as a DNA, not says as a nucleic acid. First, he was using the word that is a nuclein. The Frederick Mischer named this acidic substance to nuclein, not nucleic acids. Later it is called as a nucleic acid, but first time it is named as nuclein by Frederick Mischer. Okay. Next up, what about the length of the DNA? The length of the DNA is not same in all living organisms. It is different. Okay, the length of the DNA is a characteristic feature of all living organisms. It is not same. Suppose in human beings, the length of the DNA of human beings, the length of the DNA of bacteria, virus is not same. It is different. Right. But how can you measure the length? How can you measure the length here? The length of the DNA is usually defined as number of nucleotides or pair of nucleotides referred to as base pairs. That means in any living organisms, the length of the DNA is measured in the form of number of nucleotides or number of nucleotide pairs, nothing but base pairs. Okay, The length of the DNA is referred in the form of number of base pairs present in it, right? So it is the length of the DNA is a characteristic feature of an organism. That means it is specific, not same in all organisms. Some examples are given here. Suppose the length of bacteriophage 5174. Bacteriophage 5174. It is one type of virus. It is not a bacteria. Bacteriophage, no? Phage means what here? Virus. The length of the DNA in bacteriophage 5174 is 5386 nucleotides. 5386 nucleotides. Here we are saying nucleotides, but not nucleotide pairs, but not base pairs. What is the reason behind this? Why? Because here the DNA is single stranded. Whenever the single stranded DNA is present, we can define the length of the DNA in the number of nucleotides, but not in the number of nucleotide pairs or base pairs. Okay? Whenever simply here nucleotides is given, that should be which type of DNA? Single stranded DNA. Generally, we know that DNA is double stranded structure, but in some viruses like bacteriophage 5174, the DNA should be single stranded and it is circular. Which type of DNA is present in this type of virus? It is single stranded DNA and circular DNA. What is the length of the DNA in this virus? It is 5386 nucleotides, right? Next, it is also one type of virus, bacteriophage lambda. Bacteriophage lambda also one type of virus. In this, what is the length of DNA? See here, 48502 base pairs. 48502 base pair. This is the length of the DNA in these organisms. Here, see, once observe here, it is base pairs are given here. Base pair. That means the DNA should be double stranded. When base pair is given, the DNA should be double stranded and it is linear. Here circular DNA is present, but here linear DNA is present. These both are viruses, but the DNA structure is some different. The length of the DNA also different. Okay. Yes. Now E. coli, Escherichia coli. It is not a virus, it is bacteria. We know very well about this Escherichia coli. In Escherichia coli, the length of DNA should be 4.6 into 10 to the power of 6 base pairs. This is the length of DNA of Escherichia coli bacteria. Okay. In this, already as usually base pairs are given. No? When the base pairs are given, the DNA should be double stranded and circular DNA is present. Okay, here circular, here linear, again here circular DNA is present. Okay. After that, human beings. In human beings, we can consider the length of the DNA in two ways. One is haploid content, one is diploid content. The length of the DNA of haploid content of human beings is 3.3 into 10 to the power of 9 base pairs. Whenever comes to diploid, it is 6.6 into 10 to the power of 9 base pairs. This is the length of the DNA in human beings. Generally, we have to consider the diploid length of the DNA only. That should be 6.6 into 10 to the power of 9 base pairs. Okay. This is the length of DNA. Here two viruses are given. These two are viruses. This is a bacteria and this one is a human. Single stranded circular, double stranded linear, double stranded circular, double stranded linear. Yes. Finally, one more line is there here. DNA and RNA. Whatever DNA, both are nucleic acids now. DNA and RNA, both are made up of many subunits called as nucleotides. Okay. Nucleotides are the main subunits of the DNA and RNA. That's why the nucleotides are also called what? Monomers of DNA and RNA. What are the monomers of DNA and RNA? There should be nucleotides here. Nucleotides. Okay. Now we have to discuss about what is the structure of double-stranded DNA, how their strands are made up of. The next one is composition of nucleotide. We know the nucleotide. Nucleotide is a basic unit of DNA and RNA. The strands of DNA and the strands of RNA is made up. They are made up of many nucleotides. Clear? We know that this line. But what about nucleotide? Nucleotide is made up of which kind of things? That only we discuss now. Okay? Yes. Our topic is now composition of nucleotide. That means every nucleotide is made up of three type of components. Every DNA is made up of many nucleotides, but each and every nucleotide is made up of three type of components. What are the three type of components? First one is what? Nitrogen base. Second one is pentose sugar. Third one is phosphate group. Nitrogen base, pentose sugar, phosphate group. First one, nitrogen base. Nitrogen base. Simple. They are nitrogen containing. Nitrogen bases are nitrogen containing carbon rings. Very simple. What is the definition of nitrogen bases? They are nitrogen containing carbon rings. There are two types. Nitrogen bases are divided into two types. Purines and pyrimidines. Purines and pyrimidines. What about purines? Purines having the two carbon nitrogen rings, but pyrimidines having only one carbon nitrogen ring. Okay, that means purines are double cyclic, pyrimidines are single cyclic. Very simple idea. With the help of the diagram, we can clearly observe here the purines are double cyclic, pyrimidines are single cyclic. That only given here. They are double cyclic 9-membered rings which are having 9 positions, 1, 2, 3, 4, 5, 6, 7, 8, 9. But here single cyclic 6-membered ring, only 6 positions are there, 1, 2, 3, 4, 5, 6. Here 9, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, purines are double cyclic 9-membered rings, but pyrimidines are single cyclic 6-membered rings. But here where the nitrogen is present, why? Because they are carbon nitrogen rings, no? Nitrogen should be present in this type of nitrogen bases. So in purines, what is the position of nitrogen? It should be, see here this line, nitrogen at first carbon, third carbon, seventh carbon and ninth carbon. See here, at first carbon nitrogen is there, at third nitrogen is there, at seventh also nitrogen is there, at ninth carbon also nitrogen is there. Okay, the nitrogen present at the first, third, seventh and ninth carbons. In which nitrogen bases? In purines. Next, in pyrimidines, the nitrogens present at first and third carbon only. First and third carbon. There is no seventh, there is no ninth carbon. Why? Because it is single cyclic ring. Okay. In pyrimidines, the nitrogen should be present at first carbon and third carbon. Right. What are the examples of purines? What are the examples of pyrimidines? Once you have to see here. Adenine and guanine both are belongs to purines. Adenine and guanine. Adenine, anything. Adenine and guanine both are belongs to purines. And what about to pyrimidines? Cytosine, thymine and uracil. These three are belongs to pyrimidines. You must remember one thing here. Cytosine, thymine both are present in DNA as a pyrimidines. But what about uracil? Uracil also pyrimidine, but it is not present in DNA. It is only present in RNA. That is the difference. Okay. These four are commonly present in DNA. These two are as a purines. These two are as a pyrimidines. But in RNA, instead of thymine, the uracil is present as a pyrimidine. Okay. About this bonding, we will discuss in the next topic. Just now remember, suppose in DNA, in the case of DNA, adenine is present, guanine is present, cytosine is present, thymine is present. But in the case of RNA, as usually, adenine is present, guanine is present, cytosine is present, but thymine is not present. In the place of thymine, which one is present? Uracil is present. Okay, that is the difference. One more thing here, thymine is also called as 5-methyl uracil. Thymine is also called as 5-methyl uracil. Just remember this point. Okay, na? Right. This is about nitrogen bases. Nitrogen bases. After that, pentose sugar. Nucleotide having the second chemical component, that is pentose sugar. There may be two types, ribose sugar and deoxyribose sugar. Ribose sugar, deoxyribose sugar. Based on the type of sugars present in the nucleic acids, they are divided into two types. When the nucleic acid is having the ribose sugar, that should be called as ribonucleic acid, nothing but RNA. When the nucleic acid is having deoxyribose sugar, that should be called as deoxyribonucleic acids, nothing but DNA. See here, ribose sugar simply present in RNA, deoxyribose sugar simply present in DNA. Yes. What is the difference? What is the structural difference between these two type of sugars? This is the structure of ribose sugar. This is the structure of deoxyribose sugar. There is a little difference, small difference. Suppose see here, at third carbon OH group is there, at second carbon also OH group is there. But here also structure will be same. But here at second position, the oxygen is removing here. When the oxygen is removing from the OH group of the second carbon, now this process is called as what? Deoxygenization. Deoxygenization. If oxygen is present in the second carbon, it is called as ribose. If oxygen is removed, only H is there, this is called as deoxyribose. Very simple idea. If additional OH group is present at second carbon, the sugar is called as ribose. If there is no oxygen at second carbon, the sugar is called as deoxyribose sugar. Why it is called deoxy? Here oxygen is removing now. The process of removing of oxygen is called as what? Deoxygenization. Deoxygenization. That's why this is called as ribose sugar. It is called as what? Deoxyribose sugar. Here oxygen is present. Here oxygen is absent. That is the difference. Okay. After that, this line is very important here. Which one is the first nucleic acid actually? RNA is the first nucleic acid. There is some evidences also there. Yes, just remember RNA is the first nucleic acid or first genetic material. But what about DNA? DNA is evolved from RNA by some modifications. DNA is evolved from RNA by some modification. What are the modifications? These only. The oxygen is removing from the second carbon. Due to this modification from the RNA, DNA is developed. Okay. Here, which one is the first nucleic acid? RNA is the first nucleic acid. DNA is evolved from RNA by some chemical modifications. Very simple idea. Right. After that, phosphate group. The last component of nucleotide is what? Phosphate group. Yes, the phosphate group always present in the form of phosphoric acid. Present in the form of phosphoric acid. There should be H3PO4. Yes, this is the structure of some phosphoric acid. Okay, the phosphate present in the form of phosphoric acid in the nucleotide. What is the role of the phosphate, especially in the DNA? Phosphate provide acidic nature. Yes, we are saying that deoxyribonucleic acid, nucleic acid. Why we are calling acid? When it is acidic in nature, we are calling acid. But why the what is the reason behind it is acidic? Yes, due to presence of phosphate in the form of phosphoric acid, the DNA is become as a acid here. Okay. The phosphate provide acidic nature and as well as negative charge to DNA. Negative charge to DNA. Always you must remember the DNA is negatively charged macromolecule. What is the reason behind that? Due to presence of negatively charged phosphate. Okay, here phosphate provide two things. Phosphate having two roles here. One is phosphate providing acidic nature to DNA as well as negative charge to DNA. Okay, this is about the chemical composition of nucleotide. Each and every nucleotide having how many components? Three components: nitrogen base, pentose sugar, phosphate group. Okay, I hope you understand. We will meet in the next video tutorial. Thank you.